Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's the 8th one in the disease playlist, and you should have just come from looking at monoclonal antibodies and how monoclonal antibodies can be used in medicine to treat pathogenic infections. What we're going to look at in this video is another drug that can be used against pathogens, and these are antibiotics. Now, antibiotics specifically target bacteria, but they're an incredibly powerful weapon that we have in our fight against disease. We're also going to have a look at a few of the more commonly used medicines in this playlist. It's from the GCSE specification, and please do subscribe to my YouTube channel if you haven't done so already. Thank you. So let's talk about what antibiotics are to start with. So they're drugs that kill cells. Now that could be quite bad news because we're made of cells. So if we're taking something into our body that kills cells, we could be at massive risk. But the good thing about antibiotics is that they only kill bacterial cells. So let's take a bit of time to work out why that is, why they can only target bacterial cells. So here's a prokaryotic cell. Remember, prokaryotes is the kind of overall name for bacterial type organisms. But here's a fairly complex bacterial cell. It's got loads of additional features like the flagella that you can see on there. It's got the plasmids, the little extra bits of DNA, but we're still looking at a basic cell with the same four components as of all cells. It's got a membrane full of cytoplasm that structurally makes it a little self-contained cell. And then it's got DNA and ribosomes so it can make proteins, because remember, the proteins do everything. Now, looking at that cell though, you'll see a few characteristics on it that are definitely very different to our own eukaryotic cells. So we can target those differences with antibiotics. So if you just compare the two that I've put up there, there's some really obvious features that one has that the other one doesn't. Now think, not all bacteria have a flagella, so we couldn't target that. But pretty much all bacteria have a cell wall. So we could use antibiotics that target the cell wall. Now link this back to your understanding of osmosis and the reason why cells have a cell wall. It's to prevent them bursting. So if that cell's in a liquid or water environment, water will move into the cell by osmosis. If it's an animal cell, the water might swell the cell up until the swell bursts. A plant cell can take in the water and swell only to the limits of its cell wall, but that allows a plant cell to become turgid, so it can keep the plant upright. For a bacteria, it's just about pushing it out against the cell wall so the cell doesn't burst. It's about survival. So antibiotics could target the cell wall. If they can break down the cell wall, the bacteria would take on too much water and burst, thus killing them. Now you can also target the ribosomes in a bacteria because prokaryotic ribosomes are smaller and slightly different to the ribosomes you have in eukaryotic cells like ours. So if you make an antibiotic that can knock out those small prokaryotic ribosomes, then the prokaryote bacteria won't be able to make their proteins and they'll die, because I keep saying this, proteins do everything. If you knock out the ability to make the proteins by destroying the ribosomes, the cells are going to die. Okay. The other thing you can target is bacterial DNA. Now, not only is it different in structure, if you're looking at these, the eukaryotic cell has those linear chromosomes, just single pieces. Prokaryotic DNA is in a closed loop. So that's one thing you can target about it. But as well as that, prokaryotic DNA doesn't have proteins in amongst the DNA that help to protect it. So DNA of a prokaryote is quite different to the DNA of a eukaryote. So you can target that as well. So these are the three main differences that you can target. Prokaryotes have a cell wall, they have a different type of ribosome, and they have DNA that isn't protected by these special kind of proteins. Okay, so that's how antibiotics can work. Now, essentially then, antibiotics directly kill the cause of an illness because it's the bacteria in your body multiplying by either, remember, damaging your cells or releasing toxins. If you can directly kill the bacteria, they can't kill your cells and they can't release toxins. So you're effectively hitting the cause of the illness, which means antibiotics are a cure. Now let's take a minute now to just look at some of the more historical side of antibiotics. And we're going to look at the discovery of penicillin, which was the first key antibiotic. And most modern antibiotics are a derivative of some kind of that early penicillin. Now, penicillin was discovered by this guy. This is Alexander Fleming, who's an amazing, amazing guy. based on this key observation that he made back in 1928. And he made an observation and he actually stopped to think about it and process it. Because in 1928, Alexander Fleming noticed some mold had grown on one of his bacterial plates, like this one right here. Okay, he was actually investigating the flu, influenza, but he was growing some bacterial plates and he noticed this mold up here. This is Penicillium mold. All his bacterial colonies you can see growing on this plate. But see if you notice something that he noticed. Bacteria couldn't grow near the mold. If you look at that yellow line I've drawn on here, inside that yellow line, there's not really any bacteria growing, or the ones that are growing there are very small, very stunted. There's far less growth. So he observed that, and this was just like he was literally cleaning up at the end of a day, sorting out his bacterial plates, and he happened to pick that one up and go, oh, that's curious. It was that single moment of curiosity that made him stop and go, let's examine that. Let's do a few experiments there. That might be significant. He could have just gone, oh, weird, throw it in the bin. So a brilliant observation, a pause and some consideration there. Because what he'd inadvertently discovered is that the mold was releasing a substance that killed the bacteria, which he called penicillin, after the name of the mold that was Penicillium. And in that simple act, he discovered antibiotics, which have saved millions and millions of lives all over the planet. Most of you watching this at some point in your life have probably had to go on antibiotics. Now, it could be that without those antibiotics, whatever illness you had could have killed you. So you may not be here if it weren't for the fact that you had those antibiotics. And I would almost guarantee that pretty much everyone watching this at some point in their lives has had a course of antibiotics. Now, sadly, Alexander Fleming couldn't work out a way to mass produce this. So it took about another decade until two more scientists, these guys, Ernst Chain and Harold Florey, worked out a way to create it in mass amounts. And in the 1940s, it went into production, an American company, and they could make huge amounts of penicillin, which was one of the main reasons why, well, not a main reason, but it was certainly a significant contributor as to why the Allies won the Second World War. So many soldiers in the war died of infection. Well, the fact that the Allies had penicillin, had something that they could give to people with infections that saved their lives, means more of our soldiers were kept alive, and that's what had a significant factor in turning the tide of the war. So discovery of penicillin had an impact on something as massively global as World War II. Take a screenshot of this because this is a huge amount of information on antibiotics, but then we need to look at something that's gone wrong with the use of antibiotics. So we'll head on to that on the next slide. So we need to talk about something called antibiotic resistance. Now, this is a really great example of evolution in action. So let's just look at a couple of key points behind evolution theory to start off with. So firstly, all cells have random mutations in their DNA. So you will have random mutations in your DNA. That means you have genes, because when one gene mutates, it creates a new gene, a new version called an allele. You will have alleles of genes that mean you can make a random protein that maybe no one else can make, or certainly very few other people can make. Now, that random protein that you make may not have any obvious purpose at all. It just sits in you, totally dormant, totally pointless. But if I give you an example to try and get the concept of evolution set in your head that will then apply to antibiotic resistance, imagine if that random protein that you made was a protein that could metabolize and break down the venom that is injected by, let's say, the wandering spider. So the wandering spider, highly poisonous spider, really aggressive spider, found in the jungles in South America. Now, here in the UK, we don't need to worry about wandering spiders. They don't live in this country. They can't live in this climate. So having that protein that meant you could break down wandering spider venom, pointless. There's no reason for it because you're never going to come into contact with a wandering spider. But let's say there was suddenly a massive plague of wandering spiders in the UK. They were everywhere. They were in your home, in your car, in the shops, everywhere. And everyone was being bitten left, right and centre by wandering spiders. Loads and loads and loads of people dying. If you don't die from it, it's certainly incredibly painful. But you could be being bitten all the time by these wandering spiders. Yeah, ow, that was hurt, a little bite. But because you produce this protein, let's say it's an enzyme, that can metabolize and break down the protein in the venom from the spider, it won't affect you. You won't be killed. So suddenly, because of that introduction of that horrible pressure on survival, the wandering spiders, you've discovered the purpose of your protein. So the purpose of those random proteins would only become apparent when they are needed. You might have loads of proteins that you make that you wouldn't know if they're useful unless you're in a circumstance where they became useful. This is the same with antibiotic resistance. We're going to go through this a step at a time. So let's say I put one bacteria over there. Now, bacteria, like all cells, will multiply. Bacteria don't multiply by mitosis, remember? They multiply by something called binary fission. Either way, you're going to get loads and loads more cells. Now, let's say we don't want those bacterial cells, so we're going to treat them with an antibiotic. Now, this is antibiotic A. Notice I've labelled it antibiotic A. So if I sprayed antibiotic A all over these bacteria, I would hope it would kill pretty much all of them. Most antibiotics, you can see if you buy ones to clean your kitchen or whatever, hopefully you don't because it's bad to do, you'll see why in a minute, but most antibiotics will kill sort of 99.9% of bacteria, but that will still leave some that survive. So that bacteria over there, just like you with the protein for the wandering spider, that bacteria over there is resistant to that antibiotic because it must produce a protein that can break down the drug. So antibiotic A didn't work on that one. So now if you leave that, it will start to multiply again by binary fission. You'll get a bunch more of these bacteria. But all of those are descended from that original one that had resistance to antibiotic A. So if I now take antibiotic A and I spray it on them again, they're not bothered. They'll just carry on because they're not affected by antibiotic A. In other words, you've selected only bacteria that are resistant to the antibiotic. So you might go, okay, well, I'll change antibiotics then. I'll now use antibiotic B, a different version of an antibiotic. I'll spray them with my antibiotic B. And again, yay, it should kill pretty much all of them. But if you don't use it properly, if you don't use enough, if you don't use it often enough, some of them will still survive. Now, that bacteria that's over there, that was resistant to antibiotic A because it was one of the offspring from that original one. But that one's now just proved it's also resistant to antibiotic B. That one has now got multiple resistance, and all of its offspring by binary fission will all have that multiple resistance to 2. If I use a third antibiotic, I might be selecting now for some that are resistant to 3 or 4 or whatever. And these multiple resistant bacteria are really, really bad news. Because if that multiple resistant bacteria is a pathogen, in other words, it's a bacteria that can cause a disease in humans, then you'll be in trouble if you caught it, because antibiotics won't be able to help you. You'll have that infection, you might go to the doctors, they give you the antibiotics, you take the antibiotics, and the bacteria just smile back at you and keep multiplying because they're resistant. You take another one, they'll still keep smiling because they're resistant to multiple ones. Now, we as humans have massively overused antibiotics over the last 40 or 50 years, which means we've forced the evolution of antibiotic resistant strains of bacteria, like one you may have heard of, MRSA. Okay, MRSA is a really dangerous bacteria. It shouldn't be. You should be able to fight it off quite easily. But if you're or something from another illness, you catch MRSA, it could kill you because we don't have any antibiotics that can kill it. Okay, so this selection of resistant bacteria is our own doing because we've created this selective pressure of using antibiotics. But it's a really nice observable example of evolution. It's a really good bit of evidence that kind of adds more weight to the theory of evolution, because you can literally see generation by generation bacteria getting stronger against the selective pressure that's on top of them. Okay, take a screenshot of this. It might be useful because it's got this key information, but it's the whole process that I went through in the diagrams that's really helpful. So make some notes on this, make sure you understand it, and make sure you understand how it ties into evolution theory. Okay, stepping away from antibiotics as a drug now, because antibiotics can only kill bacteria. So they're no good for any viral infections like a cold, like the flu, or like coronavirus. Bacteria can only Antibiotics can only treat bacteria. But there's lots of other medicines that are available. But a really common type of medicine is one that treats the symptoms of a disease and not necessarily the disease itself. Remember, antibiotics kill the cause of the disease, the bacteria. There's lots of drugs that just treat the symptoms. So you'd know lots of these. For example, aspirin up above me, paracetamol over there, ibuprofen. These are all the kind of drugs that are probably in your cupboards at home that you've taken throughout your life. These three are all something called an analgesic, which means they're a painkiller. So if you've got a headache or something like that, any one of these is pretty good. They work in slightly different ways, but they're treating the symptoms, not the cause of the pain. But of course, quite often the cause of the pain isn't a disease. If you've fallen over and you've broken your leg, that's not a disease, but it's causing lots of pain. But some diseases, of course, because of the damage, because they cause sore throats or whatever, do cause pain. So you need these things to treat that symptom, because if you're treating that symptom, your body can help fight it off. These antihistamine tablets, if you're allergic to something, or you've had a bee sting or something, antihistamines can get rid of histamines, which your body produces naturally in response to an attack. But histamines can raise your body temperature, they can make you itchy, they can make your eyes watery. You release lots of histamines if you've got hay fever. So antihistamines, great drug for treating those symptoms, but they don't treat the cause. Asthma inhalers. I have asthma, so I use an inhaler like this. Again, it treats the symptoms, the closing up of my airways, or in my form of asthma, an overproduction of mucus in the airways. So they treat those symptoms, but they're not going to treat the cause, my asthma, because that's something that's a part of me. But they're all really key, really valuable medicines. And what they all do is they make you more comfortable, which helps your body to recover. Now, if it's the case of a pathogenic infection, if you've got, a sore throat because you've got a bacterial infection in your throat, then taking painkillers allows your body a little bit of peace to focus on itself, to get your immune system working, to better fight them off. If you take the antihistamines, then your body temperature can go down. It can regulate things a bit more easily, so your symptoms aren't causing you massive distractions, especially when it's something that isn't actually a disease. It's an allergy. Allergies are just when your body thinks you're under attack from a pathogen, but you aren't, but it reacts in the same way. So they can just give you more comfort, help your body to recover. Now, one really, really key thing, and this links up to some really important biology, is in the case of fever. So fever is a raised temperature. Then treating the symptom, that symptom can be essential for survival. Try and think about why. Okay, so this is your body's got infections by pathogens, you're reacting to that, your metabolism is increased to try and fight them off. Histamines in their own right can help elevate body temperature. So your body temperature is rising. That's why when you're ill, people take your temperature. If you go up to kind of 38.5 degrees, you're in serious trouble, because your body temperature, your heat, is going to start doing something to your proteins. It's going to start to denature them. Remember, we talked about denaturing when we did proteins and enzymes in the proteins playlist a long time ago. Your proteins do everything, remember. So if they start to denature, that means they kind of melt in the heat and lose their shape. They start to unfold, then those proteins aren't going to be there to do their job. Key thing, proteins do everything. If your proteins throughout your entire body, all your proteins, your enzymes, your hormones, your receptors, everything, they'll all start to lose their shape. So everything will start to shut down. This is why fever and high temperature is so dangerous, way more dangerous than the actual pathogenic infection itself, because your body's proteins go into shut down. Proteins do everything. If they've shut down, you can't do anything. So high temperature and fever is very, very dangerous. And drugs like paracetamol, ibuprofen, they can lower body temperature, increasing your chance of survival, but of course helping your body to then fight off that pathogenic infection, which in itself increases your chance of survival. So these are just some fairly common drugs and how they work, linked up to a few different parts of the syllabus. So take a screenshot of this, but a key thing on drugs is what we're going to do in the next video, was looking at how drugs are developed and end up available to be bought from the supermarket, the pharmacist, or on prescription from your doctors. So we'll head to that one next. So that video is disease 9 on drug development and clinical trials. So head to that one straight away. As always, I'll ask that you please do subscribe to my YouTube channel if you haven't done so already. Give this video a like if you found it useful, and head over to my Facebook group for a bunch of extra biology that can just round out some of your overall knowledge. Thank you.